is, from his name, you know, he's from the place called Namakkal, which has also a very powerful god, whom I like very much. And whenever he comes to India, he visits Spain these days, and we talk about our research work. We were trying to do some combined lectures together as well sometime. And he is doing very good work. He moved from Utah to University of Alabama currently, and he's also working on cardiovascular protection and cardiovascular disorders. And his work has been. Could you send me an email, please?
Oh, actually, Dr. Anu, I don't have the CV in this computer, but I sent you a link.
Okay.
Yeah, that summarizes everything.
Okay, that summarizes everything. So yeah, just give me 1 second. Yeah. So he did his MSc, MPhil, PhD from University of Madras in India, and then he's been working as a professor in various centers, and various divisions also in molecular cell pathology, biomedical engineering, cardiovascular centers, and diabetic centers. And he is a member of various societies. The American Heart Association, which is one of the biggest associations in the United States, and the APS as well. And he presents his work in a lot of conferences. I know that, and because I have met him in a conference as well. And then his work is always published in news journals and newspapers. So his lab is interested in the transcriptional mechanisms for reductive stress in the heart, and he does a lot of cell signaling work with NRF2, and he has excellent publications. So in all peer-reviewed journals, and he's a very avid researcher. I've heard a lot about him from others as well. So thank you very much, Dr. Rajshekhar, for being here with us today. And I'm sure your talk and your results and your work and your inspiration is going to be great for all the students here, faculty, and all the participants in today's webinar. From here, I ask you to take it over from this place.
Okay, thank you, Dr. Anu. That was a wonderful introduction, and thank you for starting with the my native place, which I love too much, as you said about the god as well, Anjanaya. With that, we will start our journey here. Hopefully, this is going to be for the next 45 minutes of the lecture, and then questions afterwards. So as Dr. Anu mentioned, it's a great pleasure to be here, and unfortunately it's virtual, but allowed to be there sometime in the next year, whenever it is possible. However, the topic for today is going to be, as you can see the screen, I believe. Can you see the screen, Dr. Anu?
Yeah, we can see, but you have to maximize it. It is
oh, okay. Yes.
Yeah, just get the
yes.
Correct.
So the topic for today is going to be something which is very novel, that we've been studying this for the past decade and a half in our laboratory, and it's a long journey to reach here. And this actually, I would like to connect every one of you with something new, and you may not worry about the oxidative stress that we have been going through. And a lot of literatures and a lot of science and investigators work on oxidative stress for the past 6 to 7 decades. So in the past decade, we believed that the concept of oxidative stress has some unanswered questions. Then we try to understand using a model of cardiac disease, which was originally discovered in humans. So here, the topic is, again, the redox signaling in Alzheimer's heart. You might have heard about Alzheimer's in the brain, but our lab is interested in to understand the mechanisms for Alzheimer's in the heart. Is it possible? Yes, it is possible. At the end of my talk, you might be convinced that why we call there could be an Alzheimer's in the heart. And I'm going to use a technology because just match with the topic of interest in the for this conference. I wanted to highlight some of the sophisticated techniques that we use to understand some of the cardiac functions. Many of you might have heard about the heart failure, which I'm going to talk here, but in a different perspective. For example, heart failure, you might think it is the lessened function of the heart, but I'm going to talk about its increased function of the heart also is a heart failure. So these are the novel aspects of my talk. With that, let us move on to the next slide. Okay, you might have seen this recently in the last fall, that these three individuals were awarded with Nobel Prize for their contribution, which is summarized here in one slide. So this slide won Nobel Prize. You can see there are a couple of pathways reactions, which is nothing but the debate between the normoxic and hypoxic environments in the cellular life. So what do you think about normoxia? Normoxia is always like 21% oxygen. Hypoxia is anything lesser than 21%. But it could be 21, it could be 18, or it could be 10, or it could be 5. It depends on the people who wanted to manipulate their cells or tissues with a given amount of oxygen, right? So that's what this is. So how this slide, after a long time, again, reactive oxygen species which is otherwise known as oxidative stress related work, has been recognized for the Nobel Prize, but not originally for the oxidative stress. It is all about the hypoxia inducible factor, etc. Because the one single finding that won the Nobel Prize was all about how significant the role of free radicals. So this highlighting the free radicals' role in a given life, cellular life, has very significant, very high impact in the cellular physiology as well as pathophysiology. Okay, let me move on. With that, at least we are happy that we are working in area which is very well recognized at this funding crisis these days, where you can see a lot of things that are coming against, traveling against the oxidative stress, which you will understand more at the end of my talk. So here, I would like to start with the overview. So first, I'm going to talk about heart failure. Of course, a little bit of the statistics and how to diagnose the heart failure. And next, move on to the redox stress, little bit biochemistry of oxidative versus reductive redox biochemistry, and a little bit of redox signaling, which is connected to aging and protein quality control, always. Many of you might have come across with the aging is associated with oxidative stress, protein quality control, and autophagy, and all those mechanisms are tightly regulated by redox signaling. So then we will move on to some of the mechanisms associated with what we think about oxidative or reductive redox balances or imbalances, if you will. Here we are going to use two different models. One will be to showing how the reductive stress can be proteotoxic in the context of aging. There is a model called HR120G CREAP. This was the model first discovered in humans, particularly in a French pedigree, wherein the individuals who had this sort of mutation in the gene called alpha B crystallin, which is a chaperone protein, and over the time, these individuals developed some sort of cardiac disease at the age of like 40 plus. Even though they had this gene from the embryo formation, but they did not have any issues until 40 years. So why it happens? That's what we are going to look at in detail. And we are using another model. We simply translated this human disease model with our own concepts of reductive stress and connecting to the proteotoxicity using a model called NRF2, which is, you might have known about this NRF2. Most of you must be aware of this. NRF2 is a master transcriptional regulator of antioxidants, and it regulates more than 100 or 200 genes so far discovered. And the primary role is, many people think that NRF2 is a protective factor. I do agree it's a protective factor, but its protection is required only when there is a need. If there is no offense and there is no defense, right? So if you think in such way, the NR of 2, if the NR of 2 expressed in each and every cell unwantedly, then that's going to cause reductive stress. So that's the whole story is about. So then I'm going to connect you with some of the actual relevance of this reductive stress into human heart failure, which we recently discovered in the past 2 to 3 years, and then summarize my work. Okay, let's move on to the statistics. For any talk like this, the statistics is important. As we all know that the cardiovascular diseases are worsening, #1 cause of death worldwide. And you can see that the projected total cost is going to increase to treat or to handle, to manage the cardiovascular disease. But important thing to note is that the good thing is that comparing to the past decade, now the number of deaths are tremendously decreased when compared to 10 years ago or 15 years ago. That's a great news. But then why we worry about this? So it's good that the deaths are decreased. Do you think that number of patients are decreased as well? No. Why? Because there is, you can look at this panel here. So many of us or many around us have cardiovascular diseases developed in one form or the other. But the main thing is they all have some sort of treatment, management, which is called LVAD, which is left ventricle assist device. Many cardiovascular patients around the world are having this sort of treatment in addition to other pharmacological treatments. So this is a stable device that can take care of your heart's pumping function. So basically what happens through the LVAD, it's called LVAD, left ventricle assist device. So when the left ventricle is not pumping the blood properly, the ventricle is, the apex side of the ventricle is carved out, and the LVAD is inserted and then fixed there, implanted, and it can run for years, 10 years, 15 years. There are people who have this, and that's how this is being managed. But thinking about the other side of the coin, the cost is keep on increasing because of this management, and a lot of money to be spent, and the quality of the life is impaired as well. So thinking about, a person roaming with the LVADs is not going to be so happy when compared to some others without that. So likewise, on the other hand, you see that the age is also kind of decreasing in terms of like the cardiovascular incidence usually seen in the older ages, like above 65 or 70. But these days you do see 30s, 35, 40. That is the really critical part about it. And all these things, data sets are explaining to us that we still need to work on with the fundamental mechanisms, how and what happens over the time, and why these younger individuals are being exposed to this sort of issues. So that's what our lab is focusing. And I'm going to explain this a little bit in a different way. So what do you see here? Hopefully you see that there is a echocardiography, which is mainly captured from a mouse model of human heart disease, which I showed you already, which I mentioned you already about the R120G CREAB. And as you can see that the 6 months non-transgenic mice and 6 weeks transgenic mice, 6 months transgenic mice, and 12 months transgenic mice. So you see this model. I can run through the video again to understand what we are seeing here, that the cardiac function is dramatically impaired, or I would say like is it decreased or increased. We will talk about that later. But there is, you can see in the 6 months transgenic high mice, there is a tremendous heart failure going on. And this is the low line. What is TG high and TG low are nothing but the transgenic expressed in high levels and low levels. So you see the difference. And then the last panel here, what you see is that a double transgene, which has the more copy numbers and which has the only 2 months of age, you see this thing going again worst when compared to the non-transgenic heart. So all these things, do you think all these abnormalities in the heart are associated with oxidative stress? That was the question that we thought to ask. And as you can see that gradually there is this is called progressive heart failure. So these models really evoked several questions on the mechanisms that to be studied. And we are in particularly trying to understand how this transition happening from connecting to the redox signaling and this sort of cardiac remodeling. When we call remodeling our house is very good thing, because you can remodel, you can paint the house, you can create new rooms or whatever you wanted to as you wish. But in this case, the remodeling is pathological, because this remodeling eventually affects the cardiac output. So you have seen these two models. One is the actual human mouse model, and the other one is the new mouse model which overexpressing NRF2. We all know that NRF2 is good, protective, because it can enhance the antioxidant system. And through that, you expect that all free radicals are killed. Yes, we are killing all the free radicals. Do you think that killing all free radicals is good for us? That's a major question. Think about your exercise. What we do during exercise. During exercise, we increase our heartbeat. We increase our whole metabolic pathways. Our metabolism is increased. Thereby, particularly in response to exercise, the first most thing happens is that you inhale more oxygen. So inhaling more oxygen, like several times higher levels of oxygen you inhale, which means that you're taking more oxygen. And as we all know that about 3 to 4% of the oxygen that you inhale will be converted into free radicals, meaning that free radicals are part of our life. And when you do exercise, your free radical oxygen intake is increased, and you get more free radicals. Out of the free radicals, we should get bad effects, right? But why the exercise is beneficial to all of us? Why everybody these days prescribing exercise as a medicine? You have to think about it. Please don't travel always by the traditional way. Think something out of the box. That's what the main advice I used to give to the students, particularly young minds who wanted to get into research. Think about these questions. Why are we exercising? And we say that exercise is beneficial, but we get more free radicals. How come the more free radicals are beneficial? Okay, we will get some answers while we go through the remaining slides. So this is a huge problem. So we are traveling on the opposite direction, that we need free radicals, and increasing free radical levels would be more helpful to operate the day-to-day life, day-to-day cellular signaling mechanisms, including apoptosis, cell survival, death pathways are required or driven by the reactive oxygen species. Okay, let's talk a little bit about heart failure. What is heart failure? You have seen different hearts, and you have seen that how they are functioning a little bit, if not fully. So first parameter, if you go to the doctor and ask, how does my heart work? The first thing is that they look at your echocardiography. Of course, they do a EKG, that's a different purpose, electrical remodeling, electrical functioning of the heart. But beyond that, the main thing that can tell you the structural as well as functional remodeling through echocardiography. So here there are two things, systolic versus diastolic functions of the heart. So first one is the ejection fraction. So ejection fraction is nothing but the fraction of blood ejected by the ventricle relative to its diastolic volume. So what is diastolic volume? When the heart is in the resting, like diastolic stage, before contraction, whatever the volume that holds, say for example, a heart holds 100 ml of blood. That's the diastolic volume. When it contracts, then some amount of that 100 ml is being ejected out. How much it could be, like whatever that percentage is called ejection fraction. For example, someone might have 70 ml of that blood is being ejected. That's 70%. Somebody might have 50 ml. Some might have 55. So in an average, a human being, if someone has 55 to 70%, that's a normal function of the heart. Nobody will have beyond 75% ejection fraction. If someone has ejection fraction above 75% or 80%, then you might think that, oh, my heart is working very well compared to any of my friends who has only 55 or 60. That's not true. That's not true. I'll tell you why. when your heart ejects more blood, it's working very hard. It's working very hard. How long you can work very hard like that? So that has a connecting link with its relaxation. You might have noticed some of our images. If I want to go back, for example, you can see that. You can see this is working faster, but you see this septum and the posterior wall. Here you see very closer. It's not here. Likewise, you can see the next picture also, you will know that. See how close the posterior wall of the ventricle and the septum are close to each other. So it's very thing like, when your hands comes very close and separating, the time that takes to apart is taking more time. So compared to this, you can see the next picture. You see that. Now you see that how closely they are going. So the septum and the ventricle are attaching each other and the relaxation is delayed. That's what we call as diastolic dysfunction. No one notices this oftentimes because you go to the doctor, oftentimes, I'm not blaming anyone here, but what we see here is that we go to a doctor and we get this echo done and we see, oh, my heart is like 75% working or 100% working, 80% working, which means it's a danger. It's a danger sign for the development of diastolic dysfunction in the heart. Okay, so now you know what is ejection fraction, and this is the formula to calculate the ejection fraction, and we can classify this heart failure in a couple of different ways. Actually, mainly if the systolic function, ejection fraction is reduced, then that is called reduced heart failure with reduced ejection fraction. That's one way. So this is due to systolic dysfunction. There is no worry about the diastolic dysfunction, because systolic dysfunction, if it is less than 50, for example, 40 is called stage 1 heart failure. If it is less than 40, stage 30, it's going to be stage 2 heart failure. Less than 30, then it is going to be stage 3, and less than 20 is almost a terminal heart failure. That's what this is being expected. So which is all connected to reduced ejection fraction. Whereas the other side of the coin is that diastolic dysfunction. This can be described in 3 different ways. One way is that you can see heart failure with preserved systolic function, which is preserved ejection fraction. We have a person who has normal ejection fraction, but still he or she might have diastolic dysfunction. So that's called heart failure with preserved ejection fraction. Then another thing is heart failure with increased systolic function. This is very abnormal condition because, as I told you, ejection fraction is increased, but your diastolic function is also decreased. So which mean oftentimes you might see the individual with hypertension. These days, you guys, most of you guys, because I have my own nieces and nephews, they have tremendous stress for no reasons. This is the practical life. I see it. Sometimes I get stressed out because of their stress, but there is no solution for this. These are all a lot of stress, chronic stresses are going on for without any reason, which can increase your hypertension. So the hypertension eventually can result in these sort of outcomes in the heart. We don't want to get into the personal things, but still these are true evidences that I see around. And finally, I want to touch base on the final thing, which is heart failure with abnormally increased systolic function. This is what we discovered in the past 10 years of time, because not only increased, it's abnormally increased. We do see in the previous slide, you might have noticed there are two mouse models. One is the R120G and the NRF2. The mouse model with NRF2 overexpression, having too much of antioxidants in the cardiac system, having abnormally increased systolic function. Now you tell me taking more antioxidants is good or bad. I don't know, because there is no clinical advice or clinical protocol to prescribe how much antioxidants a person, an individual is needed. We do not know now. Why don't you all think about this? I mean, this sort of questions to be answered down the road. Okay, let me move on to the mechanisms a little bit. As I already shown you, this is the slide where this is M mode, which measures the diastolic function, systolic and diastolic function, where you can see that TG low and TG high, there is a closer contraction. So you can see that the distance between the systole and diastole is very longer when compared to the non-transgenic mouse with normal heart function. This has the ejection fraction of about 50, this has about 60, and this has more than say 80% high ejection fraction. So these are the things that we need to understand about the systolic function. And the diastolic function, you can see this is measured as mitral valve parameters, mitral valve early and late filling patterns, where you see that there is a distance between these two peaks, which is early and late filling, and where you can see that that distance is increasing in the TG low mice and TG high mice. So it's too much here, which indicates that there is a huge diastolic dysfunction with increased systolic function. So one is favoring, one is unfavoring, right? So these are connected each other, because every contraction after that there is a relaxation, and contraction, relaxation. So you cannot separate these things out. Okay, let's move on to where do we see this? What is the biochemical or molecular mechanisms or molecular environment where we see these sort of phenotypes in the heart? So what we see here is that, is it oxidative stress, heart failure? Yes, people have shown, several thousands of papers have been published showing that decreased ejection fraction, which is heart failure with reduced ejection fraction, and with preserved ejection fraction, heart failure with preserved ejection fraction, no change in the thing. And what did we discover here? We see that reductive stress hearts with, you can see there is a increased ejection fraction from non-transgenics to transgenic low mice with NR up to, which is abundant antioxidants, moderately abundant and heavily abundant antioxidant. You see that there is a significant, closer to 80% of the ejection fraction. And there is a huge remodeling going on. These are the remodeling like interventricular septum and the left ventricular volume, everything. So you can see that because of the remodeling, there is an increase in the interventricular septum, and here you see the left ventricular volume is decreased. The handling power, another issue we don't understand about systolic function is that when your heart is remodeled, there is the holding capacity decreases from 100 ml to 60 ml. Then you think that all 60 ml is being pumped, which is not sufficient to circulate all your peripheral organs. Okay, this is what the histogram, like you can see that histology of these hearts, 4-chambered hearts, where you can see the remodeling, huge remodeling. This is the septum, left ventricular and right ventricular, and you see the septum, and here the septal thickness is increased, posterior wall thickness is increased. So it's not only the functional remodeling going on in these hearts, structural remodeling is also going on. Think about your pizza, think about your burger, think about your all antioxidant pills. These days, I don't know about India because I've been out of India for almost a decade and a half. But in here, I see everywhere, any shop you go, the antioxidant pills, everywhere it is there. I don't know why we need antioxidants, because our own body can synthesize its own antioxidants whenever it's needed. Okay, let's move on to the next slide. So this is for food for thought. Think about this. This is a chronic remodeling occurring in a human within 10 to 15 years of time who has a bad nutritional habits and sedentary lifestyle. If you don't produce any radicals, if you sit around, if you don't produce any radicals, and using your cell phones, mobile phones, done. Please don't mistake me if I'm dictating this as a lecture. It's not like that, but it's the impacts that I'm trying to explain you. Okay, let's move on to a little bit of chemistry of redox biology. What is redox? Redox is nothing but any chemical substance, any molecule that can form reduced versus oxidized could contribute to the redox milieu or redox environment of a given cell or an organ. So here we always go with the traditional. I would appreciate Dr. Devraj, my former PhD mentor, Dr. Devraj, in here. who introduced me to this topic, and the strength of my research is that still I am working on this topic since 1996, when I started my Master of Philosophy in the University of Madras Department of Zoology. Okay, with this knowledge that I gained, one single thing, I was able to travel up to this level, and I'm able to discover so many things related to redox, particularly on the opposite direction, which is reductive stress. So in this case, we use a lot of these things like redox ratios for a given couple, which is glutathione reduced versus oxidized form, and comparing the glutathione with malondialdehyde. And we do have any other things like NADPH, NADP, and these days you might see protein thiols, protein thiols like peroxiredoxins and glutaredoxins and thioredoxins. All these are like contributing to their nature, whether they are reduced or oxidized, or contributing to the redox makeup. And all these things are tightly regulated through metabolism. The metabolism, again, whatever the intake, which the intake is the food, and when do you take it? We can discuss a lot about circadian clock. I pretty much see current kids do not have a proper cycle. The circadian clock is messed up, so that really your system is confused about it. Okay, let us move on to it. A little bit of understanding about the redox spectrum and how to score it. Okay, let us focus on the left side panel, which is the towards blue. So here you see that it's supposed to be red. I don't know why it's blue. Maybe some transfer issue here. It has to be severe, more red here, hyperoxidative. So normally, normal setting, our cells are operated under slightly reduced level. If there is a little challenge, then you would see that there is a pro-oxidative condition, little bit, and then beyond that, it could be oxidative. While these two conditions could be acute, there is no issue. So because this normal range, all physiological signaling happening, either pro-oxidative or normal. These are the key regulators of physiological signaling mechanisms. And if it goes beyond this oxidative, yes, and goes beyond this reductive, we say there is adaptive signaling occurs. So these regions can be adaptive, mechanisms of adaptive signaling can be evoked, but the extreme, which is hyperoxidative, which is a chronic setting. But oftentimes we think that oxidative stress is the cause for many chronic pathologies. No, because hyperoxidative setting is not occurring immediately. It takes time. So it's not an acute response. It can happen if there is an intoxication, if there is a poison, if there is a hydrogen peroxide produced in your body tremendously, then that can go to this level immediately. But this will take time. This is a process. So it has its own time to reach there. So during which your system can respond and evoke the antioxidant system, priming the antioxidants to protect from this going from here to here. Whereas on the other hand, you see the hyperreductive setting. So totally what we are claiming that, hypothesizing that both extremes of the redox spectrum will be bad in the sense they could be pathological. They could be evoking pathological signaling rather adaptive response signaling. Okay, let us think one question here. One good thing is about the yes, Anu, I'm doing good.
Yeah, good.
Are we good,
Yeah,
Dr. Anu?
good.
Okay.
You can carry on. You can carry on. You have another 10-15 minutes. Yeah.
Yes. So what we have here is that the natural defense, and which has the so there is a natural defense, which I meant by the antioxidants are stimulated in response to oxidative stress. So we do not know whether there is such an anti-reductant mechanism in the system. So that's another question which is unresolved yet. Okay, so how to connect this with the protein quality control? You see there is increased aging and less protein control. Protein quality control is impaired. It's normal. And Impaired cytoprotective signaling can lead to this sort of mechanisms like ubiquitin proteasome system, autophagy, sustained ER stress, all can be connected to proteotoxicity, and which is oftentimes seen in cardiac remodeling, hypertrophic cardiac myopathy, and Alzheimer's, Huntington, and Parkinson's, etc. So moving on to the working hypothesis, these are two connected, which I already said. Aging and protein quality control and are important for cardiac health, but when it is regulated by properly redox homeostasis, otherwise this can mess up and it can lead to physiological aging becomes pathological aging and pathological cardiac remodeling, and eventually it leads to heart failure and sudden death. Okay, this is the model that I showed in RN20G, which was discovered in about 10 to 13 years ago, where you can see that a heart with cardiac hypertrophy in this model, these humans developed this over the time of 30-40 years. And this cardiac hypertrophy is hugely associated with the protein aggregates. This is what we call as Alzheimer's in the heart. You can see that the alpha-bicurstline is the mutated protein, the green color, where you can see these aggregates are perinuclear, around the nucleus. So this is the particular stain to stain misfolded proteins and aggregated proteins, and where you can see the co-localization of these, and you see that the perinuclear aggregates, big aggregates, whole cardiac myocyte is full of aggregates. So this is happening not in one overnight. It takes time, and eventually it leads to the death of these animals due to heart failure. And here is the slide where you can see that age-associated progression of the aggregation. So this is not happening in overnight. That's what I said before. Protein aggregation does not happen on day one. It progresses on aging. So where you can see non-transgenic, 3 weeks old transgenic, 6 weeks old, 9 weeks, 3 months, 6 months. At this age, these mice develop severe cardiac hypertrophy, and which is associated with good. You look at this, NQO1 is an antioxidant enzyme, and direct target of NRF2 is being upregulated, meaning that antioxidants do not help this process. And here is the evidence for how these hearts, RN20G hearts, are having increased levels of nuclear levels of NRF2, which is leading to reductive stress when compared to the non-transgenic mice. And here is the signaling mechanism, how NRF2 keep-on works. And NRF2 keep-on stays in the cytosol, but in response to stress, it can get into the nucleus and activate the NRF2-dependent antioxidant genes. But one might ask, in this system, which is how the NRF2 is activated, because there is no oxidative stress. That's right. But the issue was that these proteins that are aggregating here that you see are impairing the KEAP1. KEAP1 is sequestered into the aggregates because KEAP1 has too many cysteines in it, which makes it very vulnerable to become aggregated in these hearts. Thereby, it is leading to releasing the nuclear NRF2, which results in the reductive stress. However, moving on to some of the NRF2 dependent models, what did we do here was we created a mouse model with constitutively active NRF2 by removing this domain, which is the interacting partner with the KEAP1. By removing this, you can we were able to generate this interesting model. It's the first novel model available for studying the reductive stress, and which has the dose-dependent effects on NRF2 function as well as the NRF2 transcriptome profiling. So you can see the redox phenotyping, redox scoring has increased dose-dependently in these hearts. We call them as reductive and hyperreductive. And along with this, significantly reduced oxidation is an important thing we noticed. So there is a 4-HNE. Oftentimes you see more HNE in the oxidative stress. Here it is decreased dose dependently, and you see the DMPO adducts. DMPO is the radical that can bind to the oxidized proteins, where you can see that there is decreased signaling for these, so which are associated with reductive stress in these mice. And this is also, we can call this now is the chronic reductive stress induces cardiac hypertrophy. This is the model that we used to develop this, and what we see here in these hearts, mimicking your RN20G hearts, you can see that the heart is becoming bigger at the age of 4.5 months, with cardiac hypertrophy and the atrial thrombosis also. So this is the big issue in these hearts. We are in the process of publishing these data now under review, and also the transcriptome. Mainly, there are one of my PhD student published recently a paper on this, that redox genes, you see, they are upregulated, and protein folding genes are downregulated. So there is impairment of protein folding and increased cardiac hypertrophy. The pathology is already occurred in these hearts. So these are transcriptional evidences. With that, I would like to summarize. Summary where you have the wild type and NRF2 transgenic mice, where you can see that the either RM20G mouse model or the CNR2 mouse model, you can see that there is a tremendous antioxidants, and which results in overall it results in the GSH redox ratio is increased and decreased RYS levels that you saw, and lipid peroxidation is decreased, and everything leads to chronic reductive stress, which resulted in proteotoxic cardiac remodeling, and which finally somehow connected to the humans we wanted to, because how does this exist in the human disease? Yes. So with the help of, I would like to thank you, Dr. Sudha Ramalingam and Dr. Ramalingam from PHG Medical College, Coimbatore. I'm a faculty there, visiting faculty. I used to visit PHG Medical College every year for 1 month. And we spent time on measuring the redox profiling in these patients. And what we recently discovered was that there is a, not all patients, all heart failure patients have heart disease. That's what we discovered. Out of the patients what we did totally, there were 29 control subjects, and then there were heart failure about 50 patients. What we see is that 23 of them do not have any redox issues, which are meaning that they have normal redox. And 19 of them have, of course, oxidative, and 8 of them had reductive. So how to treat these reductive patients? Or the patients with, say, normal heart rate, sorry, this is a heart failure, but normal redox and hyperoxidative redox and hyperreductive redox. So how to treat these patients is the big question. So can we treat them with antioxidants? Of course, for the oxidative stress patients, you can prescribe antioxidants. But for normal redox, we don't know whether antioxidants or antireductants. We don't know yet what to do with these patients. But the heart failure with reductive redox must not be treated with antioxidants. That is going to be very dangerous to these patients. So we are in the process of large scale studies to understand how many of these patients are having reductive condition in their system. So with that, this is going to be the final slide to educate a little bit about how this redox work. The redox system work, Dr. Hansen and Dean Jones were my collaborators from Emory University. They have nicely explained a decade ago or even before then. So the redox state is not unique. Sorry, it's unique. It's not uniform across the board, where you can see that plasma has a different redox, cytoplasm has a different redox, nucleus has a different redox, and mitochondria and endoplasmic reticulum as well. So particularly endoplasmic reticulum compared to others has little bit oxidative redox. So this oxidative redox is needed for its function as a protein folding, and which eventually result in ER stress and leading to protein misfolding and proteotoxicity. So this is important to give a closer look on the redox state. So with that, I would like to throw this. When you are in stress, take a deep breath and relax. Why we need a deep breath? If breath is going to give you more oxygen, and that's going to increase more radicals, and that's going to be dangerous. No, it might cause reductive stress. So taking deep breath is good because it can take care of your signaling mechanisms. So with that, I would like to thank my mentors, former mentor, Dr. Devraj, and my postdoctoral mentor, Dr. T. Chandra from IIT Madras, and Ivor Benjamin from University of Utah, Salt Lake City. And of course, my folks from my laboratory, Justin, Gobinath. Justin was the one who initiated this work, and Gobinath joined later. And then now we have Dr. Kishore from University of Madras, and Dr. Sunny from Trivandrum, and my grad student. Everyone contributed to this work with the other MD students and bachelors. And we have a lot of collaborators around. Dr. Victor Daly-Usmer, well known for free radical biology, and Martin Young for circadian clock, and so many other clinicians involved in this work. And also I have other collaborators from elsewhere. As I said, the Emory collaborator, Dr. Dean Jones, and many other folks, Dr. Dale Abel, and etc. With that, I would like to thank unfortunately, we are funded with American Heart Association, NIH, and recently my postdoc got an AHA fellowship. And we do have the local funding from University of Alabama Pathology, and also University of Utah as a supporting location for me. And with that, I'm open for questions. I'm sorry if I took longer than what you expected.
No, absolutely. It's wonderful. And I'm so happy to see a lot of people that we know in common.
I would like to know how many people were there.
About 75.
Oh, good. Okay.
Yeah. So yeah. I'm going to. It was very good in the sense that you have thought out of the box, and it is a very new phenomenon that you're talking about, that free radicals are good. So I've come across a similar kind of situation when after a myocardial ischemia, people say that more than the actual infarction or distress, reoxygenation, more than hypoxia, the reoxygenation causes more damage to the heart.
Right.
So I think it is in similar lines with that. And I wish you the very best for more and more interesting work like this. It was really good. I've been getting all the questions in the chat box, and I have been
yes, I'm happy to answer questions. Questions.
But what I'm going to do is I'm going to unmute them so that they can ask you questions. So I want to give them the opportunity to interact with you directly rather than me asking.
Yeah, that would be good. I'm trying to maximize my screen here, and I don't know. My daughter is better than me here.
So I request the participants to unmute themselves, and now I think you can unmute yourself and then introduce yourself and ask your question to Professor Rajashekar. And I request all the participants who are not going to ask questions not to unmute, because the background noise will be disturbing for all of us.
Hello.
Hello.
This is Professor Prabhu from Department of Biotechnology, Sri Venkateswara College of Engineering. I hope that you may be knowing me. I did my MPhil and PhD and MSc in AC Tech. Maybe I'm your senior in Biochemistry Department under the guidance of Shamla Madam.
Yeah, I remember your name. If I could see you, then I couldn't see you here.
You would have seen me, but I don't know whether you forgot or not. Okay, anyhow, your presentation was excellent. And I also did my myocardial infarction studies using isoproterenol and the treatment by mangiferin. And most of the cases I studied only the free radical formation, which causes damage to the myocardium, and we'll be using all the antioxidants or nutraceutical for the treatment of the prevention of disease, heart failure or ischemia of the disease. And in your case, it's very what is called new hypothetical information you have given to the audience, and it causes reductive stress will cause damage to the heart. And in this case, we can modify this reductive stress by any other nutraceuticals similar to the oxidative stress, what we are proposing for oxidative stress by using different nutraceutical models.
Okay, that's an interesting question in the field. Everyone asks this because people are dealing with oxidative stress. What is the best way to treat them, right? That's true. Before going there, I would like to comment on our recent publication regarding your isoproterenol model. Of course, we did publish in a REDOX biology. It came last year, like 2019. I can send you that paper also. It might give you some insights on how isoproterenol works in the system of NRF2 signaling. as well. So, okay, the answer to your question is that the problem that we have now is we do not know the index for oxidative oxidants, for example. So meaning that we do not have a set of numbers for how much is reactive oxygen species, how much of the oxidative intermediates are a patient is having. So if we know that, then selecting an antioxidant is also important. I mean, in general, supplementing with nutritional rich foods and foods that are rich in polyphenols are known to be in enhancing the antioxidant effects. But at the same time, in terms of for the patients, when you prescribe or when a situation where you have too much of oxidative stress is going on, it requires to know what are those actual targets to be targeted. So if we know that, then we would be able to think, okay, is it a vitamin E, tocopherol, or you want to do like ascorbic acid, or you want to do something else like mito-Q if it is the mitochondria, or if you want to do like, there are so many new things, N-acetylcysteine and all those things. So everything depends on the treatment might work if we know the right target to be targeted. So that's the bottleneck in the field. Other than that, normally most of the antioxidants that we consume as food nutrients will not be a problem. That will not affect your redox balance, and it's not going to be affecting the reductive site as well. Did I answer your question, sir?
Yes, yes. He has to unmute himself. I think Abhirami, Suresh, do you want to unmute yourself and ask a question? Okay, I think.
Hello. Yeah.
Hello.
Hello, sir. Good evening.
Good evening.
This is Abhirami, and I'm a final year BTech student in Nana University.
Good.
And my question is, there is a mention about Somras, a traditional medicine, in a book by Amish Tripathi, the Shiva Trilogy series, in which they mentioned about the function of the Somras is to remove the reactive oxygen species. But they also mentioned, like in this plot, they have also revealed that because of this Somras, there were defectives like birth deformities in children, the parents who have been overused the Somras. So is there any mention about this kind of medicines, like to maintain the redox homeostasis in the folklore or traditional medicines like Ayurveda or Unani?
Okay. Yeah, very good question. I received it very clearly. Before I go there, I would like to let you know that you asked a very important question, because embryonic development requires a lot of oxidative stress, not stress, at least oxidative milieu, because the cell division, the proliferation, differentiation occurs at the pro-oxidative setting. So if you have too much of antioxidants, that's not going to work. So that's what your question is, right?
She has to unmute herself.
Oh, okay.
I think she muted herself. Okay.
Abhirami?
Yeah. I'm asking her to unmute herself.
Yes.
Oh, yeah. So the thing is, we recently published another article in Redox Biology as well, which is talking about how come you reduce or abrogate the reactive oxygen species signaling would affect the cell proliferation and differentiation. So it seems that the pro-reductive or reductive settings are tightly regulated and impairing the skeletal muscle, what's that called? Myogenesis process. Myogenesis process is impaired. But in this case, if you ask me about any Ayurvedic medicine to improve the antioxidants, right? What you said, is it improving or it's suppressing? Again, you are muted.
Yeah, I don't know, she keeps muting herself. Yeah.
Yes, sir. To maintain the balance, instead of going too low or too high.
Okay, ma. My knowledge in Ayurvedic is very limited. I'm sorry, I'm unable to answer anything from that point of view. But in generally, if you are keep active, physically active, that would help to keep up the balance. And truly speaking, not necessarily taking too much of pharmacological agents to enhance your boost up your things, because that tilts the balance in a very quicker manner. So that's the problem. So if you, for example, if you do a single bout of exercise or 40 minutes of exercise, your NRF2 is activated. That can take care of your stuff actually. So NRF2 can respond to exercise also. Any physical activity can enhance it. So I particularly, I do not know what other Ayurvedic medicines are available, but in general, most of the medicinal plants have these properties. Most of them seems to have the polyphenolics, and which might be good for to balance. But again, the balance, we do not have the measure of oxidants versus antioxidants to balance, to see what is balance and what is the the above or below the average. So that's the main area where need more research, I believe.
Abhinami, from
question.
Center for Biotechnology, University Chennai or some other place?
Yes, ma'am. I'm from AC Tech only, CBT.
So you know me?
Yeah, ma'am. You have taken classes for me.
Okay, okay. Because your name is not very familiar, though. That's why maybe you're a quiet girl in class.
Yes.
Okay.
Okay, ma'am. Thank you, ma'am. Thank you, sir.
Yeah.
That was a good question, ma.
Thank you, sir.
Okay. So the next question is from Geeta Karpuswamy. I request her to unmute herself and ask the question if she's around.
Yes, ma'am. Thank you, sir. It was a wonderful session, and we've learned quite a new things. I mean, I had never come across Alzheimer's disease of the heart. So my question is related to obesity, actually, because you said something related to accumulation of protein. So I wanted to know if Alzheimer's disease of the heart is in any way correlated to obesity. That was one of my questions. And the other question was, today's youngsters, I mean, all of us, in fact, we have a very unhealthy eating habit, eating a lot of junk food. So does that lead to increase in Alzheimer's disease of the heart? So these were my two questions.
Okay, very important and very practical questions that you asked. Thank you for those questions, ma'am. The first question about the connecting the Alzheimer's to diabetes, obesity. Yes, my colleagues in Iowa and in Medical University of South Carolina, they have come up with this new aspect. And of course, recently we got a grant to study this particular axis, that how Alzheimer's is affecting the metabolism, as well as the metabolism affect the Alzheimer's. So it seems that when your obesity is always connected to the metabolic disorder, and when you have this metabolic disorder in any form, it's a lipid or protein or anything for that matter, so the protein quality control mechanisms are beginning to impair because of too much of intake and too much of calories, which results in the too much workload for the autophagy process, ubiquitin proteasome system, and ER protein folding. So all these things are connected to the intake of proteins, right? So which are closely related to if somebody already prone to have Alzheimer's in the brain, of course, if their metabolic processes, if their routine metabolism is affected or are hugely shifted, then that can easily exacerbate the process of Alzheimer's in the heart, in the brain, which has very likely effect in the heart also, because the problem is that heart failure patients seems to have cognitive dysfunction in the brain and vice versa. But these connections are not studied. You are asking a question that NIH has recently invested money to study that. So only like few people are funded. Our lab is lucky to be funded to study this little bit of that angle, to see how finding a lot of mechanisms that are associated with cardiac Alzheimer's, not only this R120GP protein or the proteins that I show, but other proteins which are, for example, tau protein. Tau protein is being recognized in the heart tissues, particularly the atria. The atrial abnormalities are associated with tau protein misfolding and aggregation. So did you ever think about, we know beta amyloids in the brain, and we know tau proteins and alpha-synuclein, all those are misfolded. But recently, yes, there is a strong connection, but not knowing a lot of mechanisms yet. But I would acknowledge that there is a strong connection for the cardiac Alzheimer's and the metabolism, particularly the junk food, because there are so many normal pathways which are altered, not only junk foods, but when they eat, and eating always, or sometimes the intermittent eating or eating is good, people say that, but my thing is that odd timings of intake of your food is the key problem, equal problem with the different diseases. Hello, doctor, how are you?
Hello.
Hello.
Yes, sir.
So Geeta ma'am, did I answer your question?
Yes, sir. Thank you. And I'm sorry I did not introduce myself. I'm Dr. Geeta from Kamaraj College of Engineering and Technology, Department of Biotechnology. Thank you,
nice,
sir.
Ma'am. Nice to meet you.
Thank you, sir. Thank you.
Thank you very much, Dr. Raj. It was a great experience for all of us. And we wish to have more talks from your side. Once this COVID is over, I think we'll have more real talks at Anna University. Thank you very much for your time.
Yes, ma'am. Thank you so much. I really enjoyed it, and say hello to Sar as well.
Thank you so much.
I can see him in the background.
Oh, okay. Okay, sir. Bye.
Okay, ma'am. Thank you. Appreciate it.
Bye.